This is the Professor, CBMD PhD. It is Thursday, November 16th, 2023. It is 3:28 and 26 seconds, AM Eastern Standard Time. And today's research, bioengineered bacteria. So let's jump to it here. Engineering bacteria to biosynthesize intricate protein complexes. Date November 15th, 2023. Source, Tokyo Institute of Technology. As protein cages found in nature within microbes help weather its contents from the harsh intercellular environment, an observation with many bioengineering applications. Researchers recently developed an innovative bioengineering approach using genetically modified bacteria. These bacteria can incorporate protein cages around protein crystals. As this cell biosynthesis method efficiently produces highly customized protein complexes which can find applications as advanced solid catalysts and functionalized nanomaterials. However, incorporating or encapsulating foreign proteins on the surface of a protein crystal is challenging. Thus, synthesizing protein crystals, encapsulating foreign protein assemblies has been elusive. As so far no efficient methods exist to achieve this goal, and the types of protein crystals produced are limited, but what if bacterial cellular machinery can achieve this goal? The team's innovative strategy involves genetically modified Escherichia coli bacteria to produce two main building blocks, polyhedron monomer and modified ferritin. On the other hand, polyhedron monomer naturally combine within cells to form a well-studied protein crystal called polyhedra crystal. On the other hand, 24 ferritin units are known to combine to form a stable protein cage. Ferritin has been widely used as a template for constructing bio-nanomaterials by modifying its internal and external surfaces. As thus, if the formation of a ferritin cage and its subsequent immobilization onto polyhedra crystal can be performed simultaneously in a single cell, the applications of a cell protein crystal as biohybrid materials will be expanded. As to immobilize the ferritin cages in the polyhedral crystal, the researchers modified the gene coding for ferritin to include an alpha helix H1 tag, a polyhedral monomer, thus creating H1 ferritin. The reasoning behind this approach is that the H1 helixes naturally present in the polyhedral monomer molecules interact significantly with the tags on H1 ferritin, acting as recruiting agents that bind the foreign proteins onto the crystal. Using advanced microscopy, analytical, and chemical techniques, the research team verified the validity of the proposed approach. Through various experiments, they found that the resulting crystals had a core shell structure, namely a cubic polyhedra crystal core about 400 nanometers wide, covered in 5 or 6 layers of H1 ferritin cages. As a strategy for the biosynthesis of functional protein crystals hold much promise for applications in medicine, catalysts, and biomaterial engineering. H1 ferritin cages have the potential to immobilize external molecules inside them for molecular delivery. The results indicate that the H1 ferritin polyhedral crystal core shell structures displaying H1 ferritin cages on the outer surface of the polyhedral crystal core can be individually controlled at the nanoscale level, and by accumulating different functional molecules in the polyhedral crystal core, an H1 ferritin cage, hierarchical nanoscale controlled crystals can be constructed for advanced biotechnological applications. As future work in the field will help us realize the true potential of bioengineering protein crystals and assemblies, with any luck, these efforts will pave the way to a healthier and more sustainable future. And this is the Professor of Space Technology, Advanced Physics, Mathematics, Computer Science, and Leader of Research. This video was created for educational purposes only. Hope you enjoyed listening. Until next time, have a great day.